Hi everyone, this is Andy from Med School EU, and we're going to continue on with the section 2 of the IMAT for biology. And in today's video, we're going to cover the structure and function of the cell membrane. Now, before we begin our discussion with the membranes, I wanted to talk about cystic fibrosis because I find this quite fascinating. So, cystic fibrosis is one of the most common genetic diseases. It's caused by mutation to a gene that codes for a protein. And this protein is actually one of those proteins that's embedded within the plasma membrane. It is a conjugated protein, and this protein is responsible to pump chloride ions out of the cell. So it would, if this was inside the cell and it had a lot of chloride ions, the protein that cystic fibrosis is associated with, it pumps these chloride ions out of the cell like that. Now, what happens with cystic fibrosis is that it produces these proteins because of the genetic mutation. It does not pump the chloride ions efficiently outside the cell. And with this flawed function of this pump, it results in water being retained within the cells, and especially in the lungs. So a lot of these plasma membranes, they exist in the lungs, and specifically this protein exists in the lungs. And what happens is that you end up building, because the cells retain a lot of water within the cells, as the water stays in the cells, and the water kind of builds up because the ions are not being filtered out of the cell. And what happens is that there's a buildup of thick, dry mucus that cannot be effectively removed by simply coughing. So I find this quite fascinating. I wanted to begin with this. And now we're going to take a look at the specific structure and the function of this plasma membrane. We're going to begin our discussion with the structure of membranes. And the first thing we must know is that membranes are selectively permeable barriers, and it speaks for itself. Membranes are designed to allow certain things to go in and out of the cell in order to control the movement of ions, control the movement of different molecules within the cell, and control their concentrations. Now, the way the structure of membranes is depicted is by this thing called fluid mosaic model, and we're going to go over what this model is. So here we have the structure of the membrane, and we're going to label certain structures here just to depict the fluid mosaic model and what it really stands for. So first of all, this would be the plasma membrane. It is the phospholipid bilayer. So we're going to label this bilayer because it has two layers of phospholipids. These little structures are called phospholipids, and there are two layers facing each other. And we're going to go over exactly how these are formed and why they stick in this sort of arrangement. Now, these little structures within the phospholipid bilayer, these yellow structures here, they're called sterols. And sterols is another fat lipid molecule that is hydrophobic. It is water-fearing. This is why it is within the bilayer. And specifically, the phospholipid bilayer would have cholesterol, is the type of cholesterol. Make sure I spelled that right. That is the type of sterol molecule that's within the bilayer that shows up most often. Now, these little projections that stick out of the membrane, this one is called a glycolipid. And what glycolipid really means is that glyco, it's sugar, lipid is fat, and it's a structure that has parts of it being a lipid and parts of it being a sugar molecule. Now, these little projections that are sticking out of the proteins, and they're kind of floating around the membrane as well, these ones would be called the carbohydrate groups. There's plenty of carbohydrate groups that are on the outside of the plasma membranes. Now we have two types of proteins that are embedded within the plasma membranes. One type of protein that spans from the outside to the inside, it spans the whole width of the bilayer. That would be this one and this one that I depicted, and this one as well. These are called the integral proteins. So integral proteins. And typically these proteins are involved in transport of nutrients. And the other type of protein that is involved in the plasma membrane would be this one right here, and this little structure there, and these little ones popping out there. These are called the peripheral proteins. So the mosaic aspect of the fluid mosaic model refers to the fact that most membranes contain an assortment of types of proteins. As we have shown here, the peripheral, the integral proteins, and the other different chemicals and products that are all around the bilayer. This is why it is called the mosaic model, because it has all these other different aspects and different structures. And this includes proteins involved in transport and attachment, signal transduction, and processes such as electron transport. Now the membrane is typically made up of lipid, and it's made up of protein. And the proportions of both differ between the different cells. So for example, if we're looking at the inner mitochondrial membrane, which contains a large amount of protein, so about 75% protein and 25% lipid. Now, if we're looking at just the typical cell plasma membrane, not the mitochondrial membrane, but the cell plasma membrane, it would contain about 50-50, so it would just be half of lipids, 50%, and 50% of protein. And looking at the other extreme, the myelin, which is a membrane that functions to insulate nerve fibers, and it's composed just to promote electrical activity. It is composed mostly of lipids, and that's about 82% lipid and only about 12% protein. And then the other structures would fill in the gap between the rest. Now, another aspect we must consider about the structure of membranes is that membranes are fluid. And what this really means is that the lipid molecules of the bilayer vibrate, flex back and forth, they spin around along axis, they move sideways, and they exchange places within the same bilayer half. So all of these little phospholipids, they would kind of be moving around and shifting back and forth, side to side, and shifting places within the bilayer, but this mostly happens on just a single side. And if it only rarely occurs that a lipid molecule flip-flops between the two layers. And we're going to talk a little bit more what makes the membranes more fluid or more rigid based on the properties that it possesses. Now, another aspect of membranes to consider is that membranes are asymmetric, meaning that the phospholipid layer that's on top does not line up with the phospholipid layer that is at the bottom. These two phospholipid bilayers are not symmetric, they're asymmetric, and they do not correspond in an alignment with each other. All right, now let's zoom in a little bit on the lipid portion of the phospholipid bilayer. And we're going to take a look at the most basic structure of the lipid that's involved in the membranes. And that structure is called the phospholipid. And this is it right here. So this is what I drew here, and it's not a real depiction of what it looks like. However, it is a good analogy to consider all the parts of the phospholipid. And the foundation or the underlying fabric of all biomolecule membranes is the lipid molecules, and collectively the term lipid refers to a diverse group of water insoluble molecules that includes fats. And this is something that we discussed at the beginning of the course. And phospholipids are the dominant lipids in membranes. As you can see, phospholipids pretty much fill most of the membrane with just the embedded proteins inside of it. So we must take a look at the structure of it and how these phospholipid structures really form and what they really are. So first of all, looking at the head of this structure, the head would be here, and this is the biochemical aspect of it. Now the head consists of glycerol. So glycerol is this red one right here, and it is linked to a phosphate, which is further down linked to an amino group or several alcohols. Now, because of these charged molecules like the phosphate and the nitrogen here that is charged, or any alcohols that would be linked to here, this makes the head to be a polar molecule. This is the polar side of the molecule, making it hydrophilic. Hydrophilic. And what hydrophilic really means is that it's water-loving. It is attracted to water, and this is why you have the heads pointing outward to outside the cell and inside the cell, because typically inside the cell is the cytosol and the cytoplasm is mainly water, so these heads would be attracted to that, and that's why they're facing in these directions. Now, if we're looking at the hydrocarbon tails, because why it's called hydrocarbon is because it's mainly composed of just hydrogen and carbon molecules bonded together, and it is just a long tail of hydrocarbons. And because it has no charges, it is not polar, it would then be non-polar molecule. And if it is non-polar, it is hydrophobic. Hydrophobic. And of course, what this means is that it's water-fearing. And this is why you get all these little tails. They're all kind of composed in this one structure because they are experiencing hydrophobic interaction. Hydrophobic interactions. And this is something that we talked about earlier in the course. And because the phospholipid has two sides to it. It has the hydrophilic part, which is the head, and the hydrophobic part, which is the tails. Because it has these two parts to it, in general, this molecule is called an amphipathic molecule. Amphipathic molecule. And what this means is that it contains the molecule contains a region that is hydrophobic, which is water-fearing, and a region that is hydrophilic, which is water-loving. Now, if you put a lot of phospholipids together, you just kind of throw them out there, what's going to happen is that because of these hydrophobic and hydrophilic parts to the structure, it's going to form hydrophobic interactions. And we should already know about these interactions. The essence of this type of interaction is to avoid water. It's to push the water out of the hydrophobic regions. And this is exactly what happens when you have these phospholipids all lined up together. As you can see, the heads would be pointing towards the water, and the tails are all wound up into this little structure, and this is called a micelle. And the micelle is typically just the fundamental structure of what makes up plasma membranes. And plasma membranes, as you can see, they also have structures that are aligned with this physiology, making it very, making these hydrophobic interactions very known in this structure as well, because it hides away from the water in the regions that are hydrophobic. Now, the next important aspect of this model that we're going to discuss is the fluidity of the membrane, because the phospholipids are highly responsible for the fluidity of the membrane and whether it is rigid or it is fluid. So fluidity depends on three major factors. That would be the fatty acid type, the temperature, and the sterols, the presence or the absence of sterols. So first, let's discuss fatty acid type. Now, if we take a look at the structure of the phospholipid, you would notice that the hydrocarbon tail has this bend right here. The structure bends, and this is because of the double bond that's involved in carbohydrate tail. And this makes this tail and this hydrocarbon structure unsaturated. And why is it unsaturated? Because it does not have the maximum amount of hydrogen bonds that it can possibly have. Therefore, it becomes unsaturated. Now, the effect that unsaturation has on the phospholipids and the fluidity of the membrane is that the more unsaturated fatty acids of the lipid molecules, the more fluid the membrane. So fluidity would increase if we have more unsaturated fatty acids. Saturated. And more double bonds. So unsaturated fatty acids are produced during fatty acid biosynthesis through the action of enzyme called desaturases. Desaturases. Because typically phospholipids are produced saturated from the get-go. They're saturated. However, these desaturases, they would be introduced into the plasma membrane, and what they would do is they would form these double bonds within the tail, the hydrophobic region, causing it to be more unsaturated, which increases fluidity if it is needed. And of course, desaturases abundance is going to be regulated at the level of the gene transcription, so at the mRNA level. The gene is typically not going to be transcribed if the fluidity of the membrane is already good enough. If the plasma membrane is already fluid, it is flowing, and it's not rigid, then the cell is not going to produce these desaturases because it is not needed, because then it's going to make the fluid, the plasma membrane, too fluid. Now next, of course, we know that temperature affects all kinds of reactions, and of course it affects the structure of different molecules, and phospholipids are no different. The higher the temperature of the membrane is going to increase the fluidity, and this would go hand in hand with the fatty acid type, because the higher the fluidity due to temperature, it's going to decrease the transcription and translation of the desaturases, and it's going to decrease the fluidity by controlling the fatty acid type. So if you are outside and it's burning hot and the core body temperature is rising, the cells in your body are not going to produce the desaturases because it is trying to keep the membrane in its equilibrium fluidity. And finally, number 3, sterols. And I mentioned about sterols in the previous slide, and it is these little yellow dots that are within the plasma membrane, and these are also fat molecules. And typically, the best example of a sterol is cholesterol. But it does not exist in all organisms, and it's found in membranes of animal cells, but not in plants or prokaryotes. So it is very limited to just animals and a couple of other types of organisms. And sterols act as membrane buffers. So at high temperatures, if the temperature is high, they help restrain the movement of lipid molecules by reducing the fluidity of the membrane. So there's going to be less cholesterol, less sterols. And obviously, if the temperature is low, the sterols and the concentration of cholesterol is going to increase within the plasma membrane, and is going to cause the fluidity to increase because the temperature has dropped. And that's typically how the cell would regulate the structures and the fluidity of the membrane. All right, so now we're going to take a look at the functions of membrane proteins. And we're going to discuss one of them in great detail in this lecture. So first one right here, this is going to be the transport of molecules. As you can see, you got the bilayer, the phospholipid bilayer, and this is the protein. I did my best drawing a protein. There are molecules that exist outside the membrane, and what the proteins do is they basically allow these molecules to come in and fit through the membrane to come inside the cell, and that would be the transport function. Transport. Now, a lot of plasma membranes are also involved in enzymatic activity. So they're enzymes. There are enzymes that exist just floating around in plasma membranes. And what they do is they form catalysis of different reactions, forming synthesis or the breakdown of different molecules. Plasma membranes are also involved in signal transduction. And what signal transduction does is that there's a receptor on the plasma membrane, and once the chemical signal, some sort of acetylcholine, usually ACH, acetylcholine, would receive this signal, and that would be the receptor that is on the plasma membrane, and it would give off some sort of signal to the cell, either to produce a certain molecule or to break down something. And that's the function of signal transduction. And the final function of plasma membranes is going to be attachment and recognition. And that's basically molecules, different cells that would be in near proximity to each other. They're going to have some sort of attachment, and that is formed with protein structures that would form attachments within the plasma membrane that is caused by these proteins. All right, so this brings us to the end of the lecture, and in the next one, we're going to talk about more specifically about the transport across the membrane.